So thank you very much for the invitation. It's great to be here. I didn't know Svi, but I'm honored to be at this event in honor of him. Okay, so we've heard a lot about G protein coupled receptors yesterday, and they're particularly interesting because they're incredibly versatile signaling molecules. As you heard from Bob, they signal through G proteins, and often they'll signal through more than one G protein, and that can be in a cell-dependent context. So in one cell, they might signal through, say, a GS, another they might signal through GI. And they also interact with kinases and arrestins in an agonist-dependent manner, and this is a very complex system where it can simply be to desensitize the receptor or to be another signaling cascade. So if you want to discover drugs, it's challenging because you have to figure out which pathway is the therapeutic pathway and which pathway might cause adverse effects. In addition, these receptors can respond in a variety of different ways. What I show over here is essentially an efficacy diagram where we have these different kinds of drugs. But I want you to note at the beginning, many of these receptors have some sort of basal activity. And this is probably even physiologic. An example of one where you don't have any basal activity is rhodopsin. You don't want any kind of noise in the visual system. But most, in particular the adrenergic receptors, most adrenergic receptors have some level of basal activity. There are drugs that suppress that called inverse agonists. And then there are drugs that span the efficacy from weak partial to full agonists. And then drugs that are simply neutral antagonists in that they block other drugs from binding, but don't have any efficacy. And actually, neutral antagonists are very difficult to find. Almost every drug has some level of efficacy, either inverse or positive. Of note, if you want an agonist for a drug, typically partial agonists are better tolerated. So these are all considerations for the development of drugs. And since being in Bob's lab, I've been particularly interested in understanding the molecular basis for this incredible versatility. So I thought I'd start by giving you a few highlights of my LEFCO lab years, and then move on to work that I've done at Stanford. So I joined the lab in 1984, having never met Bob. I visited Duke twice. He wasn't there either time. But nevertheless, I decided to join his lab. It was at a time where Bob's lab, I don't actually remember how many people, you probably don't even know how many people. It was in the 30s. I could never, every time I tried to count, did I count him? But they were almost all postdocs. There were a few graduate students, and there was one token MD. And the previous token MD had just left. So there was a slot open, and he pretty much took you sight unseen. So I was very fortunate. That was my first stroke of luck. I'm going to talk about this in a minute. This had nothing to do with me, but it was a really important event, the purification. Then in 86, the first gene was cloned for the beta-2. 87, the alpha-2 gene was cloned. And this allowed us to do some interesting chimeric receptor studies. And then also during this time, there was the cloning of other genes by homology and not purification. But Bob mentioned this yesterday, but I still think this is an amazing story. And more so because since I've gotten into some biochemistry, I realize how truly heroic this was. So Jeff Benovic purified this protein, and by the time I arrived in the lab in '84, there was this protein sequence. So you can see it was 33,000-fold purification from hamster lungs. So 1 nanomole, or about 50 micrograms, of pure receptor from half a kilogram, roughly 1,000 hamsters donated their lungs for this effort. And from this, it was absolutely pure protein. He chewed it up and got this profile in HPLC. And these little peaks are the peptides. And it was, I believe, this peptide that was used to make several guest oligonucleotides that ultimately allowed us to get the clone from not cDNA library, but it was initially from a genomic library. And we were very lucky because we tried for about a year and a half to get it from cDNA libraries, and were completely unsuccessful. The first shot at a genomic library gave us this clone, and as Bob mentioned, it was intronless, so we were able to get the whole coding sequence from the clone. And from the sequence, we could deduce that it had 7 transmembranes, and this gave us our first clue as to what the structure might be. And it was really the first time that I thought, okay, I'd like to know more about what the structure looks like. At about the same time as I arrived in the lab, John Reagan was purifying another adrenergic receptor, the alpha-2A receptor, from 84 liters of human platelets. Now, this was just at the time when AIDS was being reported in the United States. And I remember as a resident, there were a few cases of this mysterious disease, but by the time I moved to Duke, it was even more prominent. And I know that John was purifying all this material without wearing any gloves at all. And fortunately, although he was probably exposed, never contracted. So this was another really important finding for us, because it allowed us to do some really interesting studies where
interrupt, you may not remember this detail, but the very first prep that we actually got sequenced from, we were all waiting expectantly to see what it was. It turned out to be an HIV protein.
So if you didn't hear, the very first peptide that came out was from the sequence was an HIV protein. So he was exposed. I didn't know that. That's a great story. So these were particularly good pairs because the alpha-2 receptor had a very clear pharmacologic profile. The beta-2 had from the beta-2, and they coupled the different G proteins, so you could mix and match them. And from this, we learned that the an important contributor to the pharmacology specificity was in transmembrane 7, and that that this construct, which contained TM5 and TM6 from the beta-2, the rest alpha-2, signaled like a beta-2 in terms of the G protein, but with alpha-2 selectivity. So we began to understand what domains were contributing what roles. One thing that was interesting for me was that some of these chimeras were completely non-functional, and some worked quite well, suggesting that pairing certain domains with each other, for example, let's see, chimera 4 was completely non-functional, suggesting that these two domains didn't fold well with these domains, but the converse chimera worked. So this got me even more interested in really understanding the structure at a 3-dimensional level. Now, Bob mentioned yesterday, so these chimeric receptors were all made. We synthesized mRNA for it, and we expressed them in xenobus. And the reason that we did this, well, first of all, transfection procedures were pretty new and very new to our lab. But after trying for a number of years to find cell lines that don't have beta-2, we knew that almost every cell line had beta-2. So if we expressed these in a cell line that had beta-2, it would confound our interpretation. So there were these frogs hanging around the lab. Bob had xenopus, and I learned that you could, in addition from their blood, you could actually get oocytes that you could express things in. People who are doing electrophysiology know that these are great systems for electrophysiology. So we started injecting oocytes with mRNA for the beta-2 and these chimeras, and they worked really quite well. We could do binding studies and signaling studies. The downside was you needed a lot of them, and my wife, Tung Sun, who was working with me at the time, would inject anywhere from 2,000 to 5,000 oocytes per construct to get these data. So this was those were, I think, probably the most meaningful things I did in Bob's lab. This was one of the most sort of unsatisfying for quite a while. It was cloning by homology. So I attempted to clone the beta-1 receptor. I found that there was a EQR1 fragment, 5 kilobase, that really hybridized well with the beta-2. So I created a partial genomic library. and identified the first orphan GPCR, which we called G protein G21, ultimately was found to be a serotonin receptor, and attempted to clone the cDNA for this because we didn't know if it was a pseudogene or real gene, and we wanted to see if we could find messenger RNA, and that led to the cloning of the beta 1. So it was kind of a crazy story. We had the beta 2. What we wanted to get was the beta 1. What we got was the 5-HT1A, and ultimately trying to get a cDNA for this gave us the cDNA for the beta 1. Now, this was a bit of a painful story because in Bob's lab, the most important meeting of the year was the clinical meetings and the ASCI meetings. And they had these huge plenary sessions. And I was, let me just go back. So Bob, we had cloned this just in time for us to put it in an abstract. Now, this is cloning a piece of homologous DNA with no function and no real identity. It was a pretty boring story. But Bob polished up my abstract and managed to get me into a plenary session. Now, that would have been okay, but you have to understand that back then, as you can probably tell, I'm not that comfortable in public speaking. Back then, I was a wreck. And before, this was a really important meeting for Bob, and he didn't want me to embarrass him or myself. So for 2 weeks beforehand, every day, I had to practice in front of Bob. And I could look on his face and see that I wasn't doing really well. But he didn't really tell me that. So it was the day of the meeting, and we were sitting about 15 rows up. I get up, I gave my talk, and I'm trembling, and I'm sweating profusely. And I come back and sit down next to Bob, and I'm really drenched, literally. And he leans over to me and he says, "That was the best talk you could have given." So after that, I moved on to Stanford. I left in December of '89 from Duke in an ice storm, and ended up in a very pleasant, warm San Francisco Bay Area. So when I started my own lab in Stanford, I had two major lines of research. One which I considered sort of feasible, and the other one was perhaps less so. One was adrenergic physiology, and that was really designed to try to figure out what some of these new adrenergic receptors were doing. So when I joined Bob's lab, we knew there were 4 adrenergic receptors, or at least I knew there were 4. And in fact, in my pharmacology textbook in medical school, I think there were only 2. But by the time I left, we knew there were 9 genes encoding adrenergic receptors, 3 betas, 3 alpha 1s, and 3 alpha 2s. But we didn't really know what they all did because there weren't drugs that were available to selectively activate or inhibit. So I chose to start trying to understand alpha 2As by generating knockouts. And as I said, this took about 5 years. It was a relatively new technology, and there wasn't any CRISPR back then. So this was my first, after 5 years, first publication. And I want to just blow up what our conclusions were. These data suggest that the alpha 2C is not required for normal mouse development or survival in a laboratory environment. So they had no phenotype. And it turns out that many of our mice had no phenotype until you started doubling them up. Probably this is the most striking phenotype. When you took that receptor together with the alpha 2A receptor, you knock them out. And these two are auto-inhibitory receptors. So when your nerves secrete adrenaline, nerves terminals have alpha 2As and Cs and reduce prevent further release of adrenaline. And if you don't have these receptors, you have chronic catecholamine, elevated catecholamine levels, and you ended up with mice that have heart failure. And this is sort of what the pathophysiology of heart failure in humans is as well. But At the same time, I probably was most interested in trying to understand structure. And the first task was to try to make enough of the protein. It turns out that cells don't like to express lots of GPCRs, particularly GS-coupled receptors. And it turns out that if you make a lot of receptor, this basal activity that I mentioned causes an elevated cyclic AMP, and that stops cells from dividing. So the first challenge was figuring out how to make enough receptor. By 1995, we were able to purify enough receptor for biophysical studies, and then in 98 started crystallography trials, which really didn't produce anything until about 2004. In 2005, we had our first crystals that we could see diffraction, not much. And then it was in 2007 we had our first crystal structure of an inactive state, and in 2011 a receptor-G protein complex. So this was the very first purification that we published using some tricks to modify the protein. But as you can see, having tags on the protein made it a lot easier than Jeff's job, and we only needed about 6,000-fold purification. The strategy was pretty simple. Insect cells, we had much better detergents at this time than Jeff had. We had detergents that behaved much better. And then several affinity chromatography steps, and including a functional purification. And we ended up getting purified protein that, again, we could use for biochemistry, biophysical studies, and crystallography. Now, with the amounts of material that we had to begin with, we really were kind of limited to fluorescence studies. And we ended up being quite lucky, because it turns out that when you label the receptor, you don't have to do anything to it, don't modify it at all. There was one cysteine that was particularly reactive, and it would almost be exclusively reactive. These other cysteines were somewhat buried, and these two cysteines would form a disulfide. So this would get labeled, and it was incredibly conformationally sensitive. As Bob mentioned yesterday, it turns out this is the one transmembrane that undergoes the largest conformational change upon activation. So we were, again, very lucky, and we started to be able to see structural changes in terms of changes in the fluorescence properties. One of the properties that are most sensitive, so when you have a fluorophore, its chemical environment determines both its intensity, sometimes its wavelength, and its fluorescent lifetime. So after you excite it, how long it takes to decay. And you can do a lifetime determination and get distributions of lifetime. So if you have a single, so this is a lifetime probability distribution. If you have a single peak like this, it suggests that you have predominantly one conformational state around this probe. If it's very broad, it might suggest that the protein is somewhat dynamic. But in this case, we had a single lifetime in the absence of drug. In the presence of a neutral antagonist, it looked like we were creating a more uniform distribution, but it was essentially the same lifetime. Now, we then added 3 different agonists. So isobutyrenol is a full agonist, salbutamol is a partial agonist, and dobutamine is a partial agonist. And what we saw is, one is that this distribution changed and it even got sharper. But we saw 2 additional peaks down here. And while we may have been overinterpreting this, it gave us a sense that each drug was sort of stabilizing a distinct state. For isobutyrenol, its state, full agonist, was distinct from that from dobutamine and salbutamol. And that if this was the fully active state and this was some sort of intermediate state, then even a full agonist doesn't completely stabilize the receptor in an active conformation. And these were both pieces of information that were ultimately very useful when we were trying to crystallize. So knowing this kind of information, we went back and thought about the model, which at the time, most of the behavior of receptors could be explained by a 2-state model. You had an inactive state and an active state. They were in equilibrium. Agonists stabilized the active state, which allowed G proteins to engage the receptor. However, the results I just showed you are more or less incompatible with this. It suggested there were additional states, possibly intermediate states. And many other kinds of studies that we've done since then, including additional fluorescence studies, NMR studies, and EPR studies, double electron resonance spectroscopies, confirm the fact that these are really flexible, that they exist in ensembles of states, that agonists simply increase the probability of being in an active state, but doesn't fully stabilize an active state, and that you really need a G protein or some other stabilizing protein to get an active conformation. And this is one of my favorite animations. It's a molecular dynamic simulation from Ron Dror when he was at D.E. Shaw, giving you a sense of how kind of complex and dynamic these proteins are. Well, as you know, if you want to get a crystal structure, you need a crystal, and the crystal has to be well-ordered protein molecules in a three-dimensional array. And the problem is if you have a dynamic protein, sometimes it's more difficult to form these well-ordered arrays. So just using the wild-type beta receptor with a couple of affinity tags, we were ultimately able to get crystals in 2004. And believe it or not, these are really protein crystals. And you can see they're quite small. We were fortunate to be able to be one of the first ones to use this microfocus beamline in Grenoble. And we did ultimately get diffraction spots, but you can see they're only about 20 angstroms. Try as we might, we were never able to improve the diffraction of these crystals. So again, we thought this was the major problem. We had to do something about this dynamic behavior. And you can imagine if the system is a simple two-dimensional system, if we have a very stable, uniform, conformationally uniform protein, we could represent those as hexagons, and they would pack nicely into a two-dimensional array. And what we had was a more dynamic system. We could make the protein pure, but each molecule was it could be slightly different from its neighbors and wouldn't pack as quite well together, and so we got poor quality crystals. So we had two challenges. One was protein dynamics, and the other was that these are membrane proteins, and most of the protein is buried in a lipid bilayer, or if purified, in a detergent micelle. So there isn't much polar surface for crystal lattice contacts. And in 2005, two really talented postdocs joined the lab, and they took on this problem with two different strategies. One, we developed an antibody that recognized this particularly dynamic region, the hopes of stabilizing it and also providing polar surface. And the other one was protein engineering, more or less inserting a well-behaved protein in this region. And within months of each other, we were able to get diffraction quality crystals. And these were published in 2007, one with a Fab and one with T4 lysozyme. And since then, probably the most of inactive state structures have been obtained with this strategy, either T4 lysozyme or some other well-folded protein. So that was in 2007. Our next goal was to try to get an active state. And the simplest thing was simply add agonists and see what we get. We weren't able to get any crystals with even the highest affinity agonist. So a colleague, a friend in Germany, Peter Gemeiner, made a covalent agonist, and we could show that when bound covalently, it was an agonist. We were able to get a crystal structure, but it was pretty much identical to these inactive state structures. Though bound to an agonist, it was inactive. And this led us back to the challenge is that we knew from our fluorescence studies and other studies that agonists only stabilize a relatively small fraction of the receptor in an active conformation. So we needed something that would stabilize it. We were actively trying to get a Receptor-G protein complex, but that was not succeeding. So we tried antibodies again. We tried mice, we tried chickens, and ultimately I met Jan Sterrett at a Gordon conference, and he was pushing the development of camelid antibodies. And so I thought, why not try? So we sent them protein. And the reason camelid antibodies are particularly valuable is that they're single chain only, and their variable domain is rather wedge-shaped, and so it fits in crevices in compared to a rather flatter surface for fab fragments. And this was one of the most rapid turnaround antibody projects we had. I think I sent in protein in the summer of 2010, and by November we had nanobodies, and December we had crystals. And so this was our first active state crystal structure. And the nanobody, we were able to select this nanobody by looking at the effect of nanobodies on the affinity for agonist. So G proteins enhance affinity for agonist, as Bob mentioned yesterday. And this nanobody 80, had essentially the same effect that G protein GS had. Interestingly, we immunized the llama with a ultra high affinity agonist. By this time we had found an agonist from Boehringer Ingelheim that had a dissociation half-life of about 30 hours. So we prepared the protein, immunized llamas, and while we immunized llamas with what we thought is an active state, we had a spectrum of nanobodies with an array of preferences, some including inactive states, partial agonists, and fully active states. And Bob's lab was actually the one to characterize all of these nanobodies. So we now had an active state, but we really wanted to understand the process of receptor activation of the G protein. And this project started in around 2007, right after we got the initial structure, as a collaboration with Roger Sunahara. And one of his colleagues in Michigan, Roger was in Michigan at that time, was a microscopist. And Roger suggested that we were making beautiful complex. Biochemically it was beautiful, but we weren't getting any crystals. And he suggested that we might look at it by EM. And what you're going to notice was that we had a part of the, so this is a T4 lysozyme, this is the receptor in a micelle. This is the G protein. And what you notice is that there were several different orientations of this small domain, the alpha helical domain, on the G protein. And it turns out that when you form a complex, it's nucleotide free, the alpha helical domain simply falls open and it's very flexible. And this was largely one of the reasons that we were unable to get crystals. So I'm going to spare you all of the minute details, but I just wanted to, ultimately we were able to solve this problem. But I want to give you some sense of how lucky we were to have so many colleagues that contributed to some unique reagents. So first of all, we had this ultra-high affinity agonist. It was, as I said, a dissociation rate of about over a day, and was so potent. One of my postdocs actually got a little bit on his fingers, and he had a tachycardia for several hours. We also, Roger had this brilliant idea that any kind of residual GDP might destabilize this complex. So he simply suggested we use an enzyme to chew it up. And this was incredibly important in forming a very stable complex. A couple of years before, we had started a collaboration with a detergent chemist. And as you know, detergents are really important for membrane proteins, and there is no perfect detergent. Pilsuk Che, working in Sam Gelman's lab, was making these really weird-looking detergents. And he was looking for someone to try them. And so we would try all of them to see what effects they had in these different complexes. And one detergent, MNG3, which is now commercially available, was very, very beneficial in stabilizing the complex. And we were the only ones that had access to it. So this was really fortuitous. Another reagent that we had access to was a special lipid for lipidic cubic phase. Lipidic cubic phase, at the time, couldn't accommodate really large proteins. until Martin Caffrey developed this smaller lipid, and we were able to get this lipid from Martin Caffrey. We, again, used a nanobody that helped stabilize the complex. We did some protein engineering, and then this whole project was largely guided by negative stain EM from Yurgo. And in 2011, these are the major players in this. Roger and myself, Soren Rasmussen and Brian DeVries were a postdoc and graduate student, and Andy was a graduate student in my lab. Brian was in Roger's lab. And these were some of the biggest crystals that we ever had, but they're still very small. And finally, we were able to get the structure. So at this point, we had an inactive state. We had an active state, which still crystallized it like an inactive state. And then finally, we had, this is actually quite recent, the inactive state of GDP bound GS and the beta 2GS structure. So we had many of the components that are represented in this cycle. But this was just one GPCR. And we didn't know how much of what we were learning was specific for this receptor. And as you heard yesterday, it's a very big family. It's about 800 members. The beta receptor is part of the largest group of this family, often called family A, or the rhodopsin family. In this, I think this may actually no longer be correct, but there's roughly almost 400 olfactory receptors. So we started working on additional family A receptors, including muscarinics, opioids, cannabinoids, and neurotensin. And family B, particularly the GLP-1 receptor, and a family C. And what we were looking for were receptors that couple to other G proteins. So there are muscarinics that couple to G11, GQ family, and opiates and cannabinoids couple to GI. And neurotensin couples to both GI and GQ. And for a number of years, we were able to make beautiful complexes biochemically, but simply weren't able to grow crystals. One of them, we could grow crystals of the opiate coupled to its G protein GI, but they were big, but they simply didn't diffract. So we were very fortunate to have made our connection with Yurgo Skiniotis, who was able to take all of these complexes and get structures by cryo-EM. So this is Yurgo. So within the past couple of years, all these projects, which were largely failing, had started to succeed. So this is the only crystal structure I show here of a complex. The rest in red are all cryo-EM structures. So we have a cryostructure of the mu opioid bound to GI, a cannabinoid bound to GI, a M1 muscarinic receptor bound to GQ and 11. So we have all the major families represented. We also have a family B receptor, and most recently a family C. And I'm going to briefly talk about these. So I first want to go through these three, which are the most closely related in the family A. And I'm going to talk about three regions of the receptor. The binding pocket, this transducing domain, which has a conserved proline in TM5 and a phenylalanine and proline in TM6, and this polar region where there are this NPXXY sequence. So the binding pocket, as you'll see, is different for all of these receptors, but it ends up having the same sort of conformational changes when it gets to the cytoplasm. So here are the three receptors, adrenaline and iperoxo. So this is an acetylcholine receptor. This is a super agonist for this muscarinic receptor, and this is an opioid. The binding pockets here are largely polar interactions with these flexible ligands, and the binding pocket here is largely nonpolar interactions. The major change here is in transmembrane 5. Here it's in transmembrane 6, and here it's in transmembrane 3. So these agonists all change the binding pockets, but they're all different for all these receptors. Nevertheless, they end up propagating to the same structural change on the inside, which is an outward movement of TM6. I want to then just mention this interesting region where you have this transducing portal. And for the beta-2 receptor and for the opiate receptor, it has this cluster of relatively conserved residues. So I'm just showing 4 transmembranes to make it simple, but highly conserved proline in TM5 packs against a phenylalanine in TM6 and an isoleucine in TM3. And this packing arrangement is quite tight and does not accommodate this inward movement. As a result, it's rearranged. And as a result of this rearrangement, you have a rotation around this phenylalanine. And that rotation is enough to be amplified into a roughly 14 angstrom outward movement of TM6. Now, so this is a really important mechanism of transduction for the beta-2 receptor. And I'm going to step back and now take a little side story about an allosteric modulator. So this, I just showed this is important for the beta-2, and Bob mentioned yesterday about allosteric modulators. The reason you want allosteric modulators is because they take advantage of differences between receptor subtypes. So if this is a beta-2 colored in blue, regions where it differs from beta-1 are in yellow. So this is the outer surface. This is the extracellular surface. If we crack the receptor open and look at the inner surface, you can see there's a lot of yellow in the extracellular vestibule, but all the rest is blue. So the binding pocket the orthosteric pocket is virtually identical. Now there's one example of the highly selective drug. So this is a non-selective antagonist. And this is a selective agonist, salmeterol. And it takes advantage of this extracellular vestibule. And this is the selectivity determinant. Okay, so why am I talking about this? Well, we tried to use docking to find an allosteric modulator that bound in this extracellular vestibule. For muscarinic receptors, this extracellular vestibule is known binding pocket for modulators. So we did the docking, Brian Scheukett's lab, and we get a hit. It's pretty weak, but you can see as you add more and more of this we call BRAC1, the beta-arrestin recruitment decreases. By making a number of analogs, we found that simply adding a bromine substantially improved the efficacy of this compound. So now 30 micromolar almost completely suppresses the arrestin recruitment. So we want to know where in this pocket it's binding, and we were able to get a crystal structure, and it wasn't binding there at all. It was binding here. And why is that interesting? Because this is right at that highly conserved proline. So this is this we call the PIF motif. And this is the region of the receptor that moves inward upon activation. So this is inactive, and activation is in gray here. So active, inactive. And what this compound is doing, it's binding and stabilizing the inactive state of this important conformational hub. Okay, so we call this a docking miss, but it's an interesting compound. So is this universal, this triad of proline, isoleucine, and phenylalanine? Well, it's true for the beta-2. It's true for the mu-opiate receptor, same sort of arrangement. But the muscarinic receptor is not. It has a valine instead of an isoleucine. And you can see the arrangement really doesn't change going from active to inactive. So it turns out there are roughly 90 family A GPCRs that have this feature. And the remainder don't. And I just looked up of those that don't, they all tend to be those that have the class B behavior that Bob mentioned. So the vasopressin, the angiotensin receptor, neurotensin receptor, muscarinic receptor bind tightly to arrestin. and they often have ligands that bind, that are biased. So this suggests that maybe these are conformationally even more versatile. And I want to show you just some data that in fact supports that notion. These are studies using NMR spectroscopy, and I'm not going to go into the details, but you can do NMR spectroscopy on GPCRs if you use selective probes. So we found that the epsilon methyl of methionine can be really useful reporter of conformational changes from within the transmembrane bundle. So this is an NMR spectrum of the M2 receptor with all of the methionines, and it's kind of a messy spectrum. You get rid of some of them that where the spectrum doesn't change much, and you end up with 5 probes that range from the outside to the inside, and you can begin to look at the fingerprints of these of these structures. And so we looked at the results of binding to various antagonists. So these are all inverse agonists. This is an allosteric antagonist, and these are agonists that have different behaviors. So this is acetylcholine. Acetylcholine would be considered the native hormone, so we call this 100%. Iperoxo is a superagonist for arrestin recruitment. Iperoxo and its related compound PR15 is a superagonist for G protein, but compound 15 is only a full agonist. So we have interesting spectrum from inverse agonist to full agonist and superagonist. And to cover this very quickly, every ligand stabilizes a distinct spectrum. There is no ligand stabilizes the same spectrum as the apostate. Even the antagonists all stabilize unique spectrum. And what was particularly surprising is agonists, the full agonist and the superagonist. So the black is apo, no ligand. Acetylcholine causes the shift field. And then when you bind it to a G protein mimetic nanobody, it shifts in the opposite direction. Superagonist iperoxo causes a different shift upon binding. And when you add the nanobody, it shifts in a completely different direction. So this is as if there are 2 different active states for these different compounds. So it suggests that receptors with a looser packing in the middle of the receptor may adopt even more conformations on the inside. Now, the one receptor that we've looked at that's easiest to explain how the ligand works is the cannabinoid receptor. And the cannabinoid receptor is one of these that's even more unusual than the M2 muscarinic receptor in that it doesn't even have a proline in TM5. So almost all family A's have this proline in TM5, and the cannabinoid just has a straight rod. And as a result, it's very loosely packed. You can see that beta 2, and here's the same region in the cannabinoid. So we were able to get a structure of the cannabinoid receptor bound to Fubinaca. So this is the super agonist that's been found in laced marijuana cigarettes, where some people have died and some people end up in the hospital. And this is the Fubinaca binding site, and it's easy to see why this agonist works. This is the most direct effect that we've seen. So this is transmembrane 6 in the inactive state, and it's this highly conserved tryptophan is held in this inactive position by a phenylalanine. What Fubinaca does is it binds and it stabilizes this phenylalanine in a different rotamer, allowing this tryptophan to rotate outward. And this small rotation in the middle of the helix with this proline right here leads to this outward movement. So this is a very simplest mechanism. We've never seen anything quite so easily understood. This tryptophan is called the rotamer toggle switch, but this is the only structure where I've ever seen it really make a difference in terms of the conformational change. Okay, so all of these receptors and complexes really behave very similar to the beta 2 in that small structural changes on the outside are propagated in different ways to the inside, and that leads to an outward movement of TM5 and TM6, and allowing the G protein to engage the receptor. And this is pretty much very similar for all the receptors that I just talked about, with some minor deviations in the interactions. As an example, here are two GI-coupled receptors. You can see that the G protein is the same, but the way they engage TM5 and TM6 is slightly different, and it's even different for the G protein, GS-coupled receptors. What has been disappointing is that although we have complexes for each one of these G proteins, we still don't understand what makes a receptor prefer a particular G protein. We're beginning to learn why a receptor won't couple to a particular G protein. For example, the mu opiate will not couple to GS because primarily its TM5 and TM6 can't move out far enough. It's only about 9 angstroms where you need a larger outward movement because GS has a more bulky amino acids at the end. But for the most part, we don't understand coupling specificity. And it doesn't really help going to another family. So we next tried family B receptor. GLP-1 is an important target for diabetes. Again, this is a cryo-EM structure. And this is a peptide receptor that has a two-step binding mode. The peptide initially engages a flexible N-terminus, and then it docks it into the core of the receptor. You can see it's very similar to the beta-2, another GS-coupled receptor. The way that the domains interact are almost identical. The major difference is that there are much larger changes in the binding pocket as a result of fitting in this large peptide, and the change in TM6 is even larger. It's sort of a gradual bend with the beta-2 in TM6. It's a complete break in the helix, and it's because of this highly conserved PXXG motif, which is found in all family B receptors. So they look very similar. If we look at the sequence of GS shown here, this is the C-terminal part of GS that binds, engages the receptor. And we look at the amino acids in the GLP-1 and the beta-2, there's really no conserved set of amino acids. Their interactions are all different. So again, we're not learning anything about coupling specificity. So to summarize what we've learned so far from these complexes, nucleotide-free complexes are similar for family A and B, except for our larger outward movement of TM6 in family B. There's a smaller outward movement in GPCR, GI, and GO complexes. Structural changes in GS and GI upon activation are very similar. They all undergo this large movement of the alpha-5 helix, and the alpha helical domain becomes flexible. GPCR G protein complexes do not fully explain coupling specificity. And then, of course, the answer is going to be yes. Is coupling specificity determined by an intermediate state? And we think so. I don't know, I can't really say yes yet, but I can provide some evidence in the next part of the talk about for an intermediate state. So we think that so these were states that we knew biochemically, receptor bound to an antagonist, the apo receptor with dynamic transmembrane 6, agonist bound receptor where we increase the probability of being in an open state, the nucleotide free complex, and then after GTP binds, it returns to the agonist bound state. But what I'm going to show you is evidence for at least two additional complexes, intermediate states that might contribute to coupling specificity. So evidence for this are some single molecule studies that I'm going to talk about. We've also done pulsed deuterium exchange studies to look at complex formation, and we we see that the state that would be represented by our crystal structures takes many, many minutes to form, much longer than it takes to undergo a cycle. So we think this state is perhaps somewhat unique to the nucleotide-free state. We also have a structure of the beta-2 bound to just the GS peptide that is different orientation than we observe here. And then finally, I didn't show this, but the NTS1 GI complex, we actually see two different states, two different complex formations. This may be painful for you to listen to, but it's one of my favorite studies, so I'm going to tell it anyway. This is a project done with Scott Blanchard and Glenn Gregario in his lab, and Matt and Dan in my lab. And I've been really just fascinated by protein dynamics. And one of the limitations of NMR and even fluorescence is there are ensemble studies. And we started collaborating with Scott to try to get more information about single protein dynamics. And this still is very much a work in progress. But what we did is we labeled the receptor with two probes, one at a reference point in TM4 and one in transmembrane 6. And we're simply looking at fret between these two probes. In the inactive state, the probes are close together, so we have high fret. In the active state, where TM6 moves out, we'd have lower fret.
How did you accomplish it? How were you able to tag on one molecule two different?
Yeah.
Because it's not so trivial, I guess, right?
Right. So this cysteine is incredibly reactive, and this cysteine is partially buried in the micelle. So we can easily label this one at first. Yeah. And these studies were done using Prism Turf, immobilizing the receptor in a couple different ways, which I'll mention in a minute, and simply recording donor and acceptor of many, many molecules. So the immobilization strategies are shown here. We immobilize a passivated surface, on a passivated surface, a anti-flag Fab, and this binds to the N-terminus of the receptor. So we can then float in G proteins, and we can see the effect of agonists in G proteins. We can also immobilize the G protein. Now, in this case, the G protein is not labeled, so the only time we'll see a receptor is if it's complex with the G protein. And this allows us to look at receptor G protein complexes in different states of GDP or GTP. And when the receptor falls off, we lose fluorescence either because of photobleaching or because it dissociates. So this is an example of the kinds of data that we get. These are single molecule traces. And in this experiment, we have receptor immobilized and G protein in solution. And you can see this is a fret trace. You can see it flickering on and off because it's coupling and uncoupling to the G protein. You can then create heat maps from many traces, and you can also represent them as histograms. So this would be the apo state. This is what we call basal activity, basal coupling. An inverse agonist, now we have high fret because it doesn't couple to the G protein in the presence of an inverse agonist. And then in the presence of a full agonist, we have just tonic low fret because it's all coupled and the two domains are far apart. Just summarizing the extremes, inverse agonist gives us the highest fret, and receptor G protein gives us the lowest fret. And agonist alone, you can see it gives us lower fret, but still nothing compared to receptor G protein. So in these experiments, that's all in the way of background. We're trying to look to see if we can find different states of a receptor G protein complex. So we immobilize the G protein, and we add receptor. So this is nucleotide free, and then we add either GTP or GDP and look at what happens. So in this experiment, we are adding, this is an epinephrine and adrenaline-bound receptor coupled to G protein. So it starts at low fret. And I'm just going to, and we add, at this point, GTP. And you can see immediately it goes to higher fret, but it's still visible. That means it's still coupled to the G protein. So we have some receptor that is clearly structurally different because the fret state is different, but it's still coupled to the G protein. So this was a state that we didn't see before. Now, in order to look at assembly, so this is disassembly. In order to look at assembly, we're looking at more or less the reverse. We're adding GDP. So we start with a nucleotide-free complex, and we add GDP and go this direction. So again, we start with a low fret. Now we add a GDP, and you can see we have this intermediate fret state that then ultimately goes to the high fret state, and we lose the signal, suggesting we have 3 different complexes. So we have the nucleotide-free, we have some sort of intermediate state where TM6 is still moved outward, and then we have a state where TM6 is just in the same state as the agonist-bound state, but it's still bound to the G protein. And if we do this for a partial agonist, you can see we have a similar effect, but partial agonist is much less effective at stabilizing this intermediate state. Now, because we're mobilizing the G protein and we want to make sure we're not creating some sort of not dysfunctional G protein, we did the same experiment but immobilized the receptor and then add G protein. Now, in this case, what we're looking for, can we reproduce these intermediate states? So again, it's receptor, agonist, and G protein, or partial agonist and G protein. And you can see we're going between this high fret state, low fret state, intermediate, and flickers back on and off over the course of the experiment, suggesting that we're seeing these states go back and forth. And in the weaker partial agonist, we see the same sort of states, but much less of this intermediate state. So we think if we really want to understand coupling specificity, we need to know what this looks like, and maybe even what this looks like. But as you can see, they're so transient that these aren't going to be captured by crystallography for sure. And it's going to be a challenge to capture them by microscopy, but this is something we're really interested in doing. So again, there's many reasons why we don't think our crystal structures are necessarily telling us everything, this whole story about activation, and there's still work to be done to try to figure this out. My last story I want to tell you about is family C. Now, family C have been more challenging, and we heard yesterday some questions about dimers. And so first of all, family C includes a small cluster of receptors, including metabotropic glutamate receptors, GABA receptors, the taste receptors you heard about yesterday, and calcium sensing receptor. And our subject has been the mGlu-R5. It's mainly postsynaptic. It signals through GQ, and it's important in a number of central nervous system functions. So this is a cartoon illustrating the structure of a family C. So this would be the 7-transmembrane domain. This would be essentially the component of a family A receptor. There's then this very large extracellular venous flytrap motif that contains the orthosteric pocket. So glutamate binds here and causes a closure of this that rearranges this connecting domain called the cysteine-rich domain that then presumably rearranges the transmembranes. There are known positive allosteric modulators that bind to the transmembrane domain as if it were a family A. And the binding pocket is very similar to family A receptors. So positive modulators bound here. Orthosteric, and negative modulators as well. Orthosteric agonists bind here. And we were able to find again from llamas, a nanobody that served as an allosteric agonist. So this nanobody, called nanobody 43, this shows the effect of the nanobody in orange here on signaling in cells that has media that probably contains some glutamate. So it's very hard to get rid of glutamate. So it's probably acting as an orthosteric agonist here. So nanobody 43, CDPBB is a positive allosteric modulator that binds here, and that also looks like an agonist, or an orthosteric agonist. Glutamate, the native agonist, and quisqualate is a very high affinity agonist. So these are all compounds that we're going to be using. This just shows what happens if you can get rid of all the glutamate. Now you see that nanobody 43 doesn't have any effect, doesn't signal, and CDPBB has a small effect, but glutamate and quisqualate still have a substantial effect. So again, this was done by cryo-EM with Yorgos Giniotis, Antoine Cole in my lab as a graduate student. And we were able to get an inactive state and an active state. And the inactive state, you can see the transmembranes are separated and they come close together in the active state. A bit more in detail. So this is the nanobody. And you can see nanobody stabilizes this arrangement of these 4 helices in an active conformation, allowing the sort of inward movement of the cytoplasmic domain. So it's interesting that the nanobody is probably largely interacting with this acidic residue at the top of the venous flytraps. And this is the inactive state. And then what happens to the transmembranes? So in the inactive state, they're separated. In the active state, transmembrane 6 and 6 interact with each other. And this just sort of summarizes that the nanobody is able to stabilize the close conformation, and this allowed us to get a stable state for structure determination. And these are the conformational changes in going from the apose state to the active state. So how important is the dimerization? Well, based on our structure, it's only 4 angstroms, so we're able to place the helices, but we don't really have a lot of detail in terms of side chains. But we created a cysteine disulfide here, which is very similar to what has been done in a glutamate 2 receptor by Jean-Philippe Penn's group. And by simply stabilizing these by cross-linking, you create a receptor that has very high basal activity and can no longer be activated by glutamate. However, you can inhibit this high basal activity with an inverse agonist or a negative modulator that binds here. So how does this connection between the extracellular domain and these flytraps work? I mean, looks like a long, flexible linker. Well, it's actually not as flexible as it looks. What we found is that the extracellular domain 2 engages the CRD and acts as a fulcrum. So when these move inward, they push on extracellular domain 2 and force the 2 transmembranes together. And that leads to activation. So to test this, we simply removed 2 amino acids at the end of this loop. And by removing those 2 amino acids, the receptor, as an intact receptor, would no longer signal. So this is a wild type. This is a deletion mutant. However, if we took the same receptors and cut off the N-terminus and just expressed the transmembranes, it now signaled normally with a positive allosteric modulator. So this is a key mechanism for moving and activating the receptor. We still would love to get a receptor coupled to G protein, and this is something we're working on now. So there's still a lot more work to be done here. So I'm going to end here and thank everybody from my group. The last story I told you about was by Antoine Cole, and all of these people contributed to the various structures that I talked about. Also, a great collaboration with Roger Sunohara and Yorgos Skiniotis, who is now at Stanford. And I thank you for your attention.